me some time. Please make my line. You know I'll answer. Call me some time. Please bang my line.
Effects of the environment can affect physiological processes such as hormones, neurotransmitters, but mainly the human brain. Therefore, the relationship between the environment and physiology is often said to be bidirectional, which essentially means functioning in two directions, where environmental enrichment changes the cerebral cortex. Therefore, the brain changes our experiences and behavior. So, what's brain plasticity? Brain plasticity, also called neuroplasticity. However, brain plasticity is a common term used by neuroscientists, referring to the brain's ability to change at any age for better or worse. As you would imagine, this flexibility plays an incredibly important role in our brain development or decline, and in shaping our distinct personality. The brain has the ability to reorganize itself and form new connections between neurons, and it is stimulated by the environment. Plasticity occurs every time something new is learned. Brain plasticity is explicitly shown after brain injury, when the brain reorganizes and forms new connections with healthy neurons to compensate for the functions of the damaged area. Connects to the question because this is an example of a study which investigates the effects of a deprived or enriched environment on neuroplasticity. Is an experiment conducted by Rosenzweig and Bennett, 1972. The aim for this study was to investigate the effects of a deprived or enriched environment on neuroplasticity, in particular, the development of neurons in the cerebral cortex. Method: lab case study. The rats were observed for a period of time in a lab setting. Different aspects of the rats' behaviors were recorded. Purpose: in order to effectively determine the results of rats' brains based on their environments, it was necessary to place them in controlled, lab environments. Case studies provide researchers with a significant amount of information. They can control the environments if desired, and more aspects of the subject's life can be observed. The independent variable was the type of environment that the rats were exposed to. Stimulating environment contained interesting toys. Deprived environment had no toys. The dependent variable was the weight of the rats, the brain, showing the amount of brain plasticity that occurred in the rats. The rats were also separated and exposed to the two environments for 30 to 60 days before being euthanized. 3 male rats were chosen from each litter. They were then randomly assigned to one of 3 conditions. 1 rat remained in the laboratory cage with the rest of the colony. Another was assigned to the enriched environment cage, and the third was assigned to the impoverished cage. There were 12 rats in each of these conditions for each of the 16 experiments. The 3 different environments were the standard laboratory colony cage, several rats in an adequate space with food and water always available. The impoverished environment, a slightly smaller cage isolated in a separate room in which the rat was placed alone with adequate food and water. The enriched environment, 6 to 8 rats in a large cage furnished with a variety of objects with which they could play. The rats were allowed to live in these different environments for various periods of time, ranging from 4 to 10 weeks. Following this differential treatment period, they were humanely sacrificed so that autopsies could be carried out on their brains to determine if any differences had developed. Results 1. The anatomy of the brain was different for rats in the EC and the IC, and also the brains of EC rats had increased thickness and higher weight of the cortex. EC rats had developed more acetylcholine receptors in the cerebral cortex, important neurotransmitter in learning and memory. Evaluation. In response to this possible criticism, further experiments were carried out, wherein the impoverished rats were handled as much as the other two groups, and the differences were still found. Also, sometimes the rats in the enriched environments were not handled, but still they retained the increased brain growth. Rats in the stimulating environment had a thicker cortex and heavier frontal lobe, associated with thinking, planning, and decision-making, compared to rats in the deprived environment. This may have resulted from the exposure to the toys in the stimulating environment, which helped to develop neural connections in the rat's brain. This is connected to the question because it's a study supporting the effect of mirror neurons as a result of observing people's behavior is conducted by Jacoboni, 2004. The methods were participants were asked to watch films of people reaching for various objects within a T-time setting and to look at human faces while in an fMRI. The participants also had to imitate the faces shown, and then they watched as the faces were shown again. Results 1. The anatomy of the brain was different for rats in the EC and the IC, and also the brains of EC rats had increased thickness and higher weight of the cortex. EC rats had developed more acetylcholine receptors in the cerebral cortex, important neurotransmitter in learning and memory. Evaluation. In response to this possible criticism, further experiments were carried out, wherein the impoverished rats were handled as much as the other two groups, and the differences were still found. Also, sometimes the rats in the enriched environments were not handled, but still they retained the increased brain growth. Rats in the stimulating environment had a thicker cortex and heavier frontal lobe, associated with thinking, planning, and decision-making, compared to rats in the deprived environment. This may have resulted from the exposure to the toys in the stimulating environment, which helped to develop neural connections in the rat's brain. Connection of study to question. This study demonstrates the effect of observing experienced actions on mirror neurons as the participants' mirror neurons were activated when they observed and imitated facial expressions. In conclusion, the two discussed effects of the environment on physiological processes are the enrichment on brain plasticity and observation of experienced actions on the activation of mirror neurons. The neuroplasticity is the brain's ability to reorganize neural pathways, physiology, based on new experiences, environment. The interaction occurs when the enriched environments affect the neural connections in the brain by reorganizing the neural connections in brain areas related to functions that are required in the enriched environment condition. Mirror neurons in the brain activate physiology when we perform an action and observe someone else performing that same action, environment, and therefore mirror the behavior of another individual. MN makes us feel as though we are performing an action by just observing it. We experience an inner imitation of the action. The relationship exists when observing another person's behavior in the environment, which therefore generates a response from the neurons in our brains, which are physiological processes of behavior. Therefore, as a result of the supporting studies and theories that a bidirectional relationship exists between the environment and physiological processes, forming a strong interaction. And